Well, welcome everyone to today's Safer Road by Design webinar. And today we have Andy Forreryd from Senzagen, an innovative company in Sweden that's been working on developing novel assay technology to evaluate skin sensitization. And Andy's theme today, I think, is a very important one because we need to use NAMs in the context of applying it to risk assessment, and especially, if possible, quantitative risk assessment to really make it valuable in practice to companies making decisions on ingredients and concentrations in product. So with that, I'm going to hand over to you, Andy. Just before I go, I'll come back at the end. Please remember there's a question box over on the panel. Think of your questions and submit them, and we'll have a good discussion at the end. Over to you, Andy, from Switzerland to Sweden.
Yeah, brilliant. Thank you very much, Barry. We don't have the similar good weather in Switzerland as here in Sweden, I can tell you. But thank you again for the kind invitation to present at today's webinar. And I would, of course, also like to take the opportunity to welcome everyone to today's webinar. So I will just briefly close down my webcam, and I will put it back on for the Q&A session later on as well. So the aim of my presentation today is to give you all an introduction to the GARD technology platform and to describe how we use a combination of genomics and machine learning in order to develop next generation of in vitro assays for skin sensitization testing. So as the title may suggest, the major focus of today's presentation will be on our most recent development of the GARD technology platform, which is called the GARD skin dose response assay, which is a novel method for quantitative assessment of relative sensitizing potency. So as the major focus of today's presentation will be on the capacity of this GARD platform to provide potency assessment, I wanted to start this webinar with a very brief overview describing why there is a need for in vitro assays that are capable of providing accurate and reliable potency information. So first of all, potency is often required by regulatory subunits, such as, for example, the REACH registration. And here in this regulatory context, the potency information that is often required is restricted to the CLP categories, Cat 1A and Cat 1B. So in addition to the regulatory requirement, potency information is also required for risk assessment, and in order to establish a safe dose of a safe concentration to use within products. And in this context, it is not often sufficient with the categories in the GHS system, but here you would require more quantitative assessment of potency on a continuous scale. So the challenge today is that the OECD test guidelines are not being developed or validated for potency assessment. They will only be validated for hazard assessment. And we can also see that it is really difficult to use these assays, even when they're used within defined approaches, in order to provide potency information. So the data in the figure here to the right summarize the data from a selection of defined approaches that we use to predict the CLP potency categories. And you can see that accuracies from these were only about somewhere between 55 and 69% in accuracy. So the GARD assay basically comprises a set of different assays that can be selected dependent on the purpose of the testing and also dependent on the amount of potency that you would require. So the GARD skin assay was the first method that we described. It was developed in order to perform binary hazard classification of skin sensitizers. And it can also be complemented with the GARD potency assay, which can be used to further subcategorize identified skin sensitizers according to the 1A and 1B categories defined by the CLP system. So the GARD skin and the GARD potency assay were developed in order to comply with the potency information that is required by current regulations. And the information from these assays can also be used for regulatory purposes. And I will come back to this later on in this presentation. But we also, of course, acknowledge the fact that although that the potency information provided by the GARD potency assay is what is often required by regulators, it is not sufficient for being able to perform a quantitative risk assessment. So for this purpose, we also decided to develop the GARD skin dose response assay, which are capable of providing a more continuous prediction of relative sensitizing potency. So before moving on to the GARD skin dose response assay, I just wanted to introduce you to the GARD technology platform, which is based on human cells in combination with genomics and machine learning. So when we talk about the GARD assay, we normally talk about a technology platform that utilizes the same modular framework in order to develop our assays. So all of our assays basically comprise of a biological system that is relevant to the toxicological endpoint that we try to predict. On top of that, we use a training data set of chemicals with known expected outcome. We use these chemicals in order to expose the biological cell system. We measure the complete transcriptome of the cells, and we identify the most potent set of predictive genes. And this is what we refer to as genomic biomarker signatures. So in the final step, we use a prediction model that is based on machine learning in order to interpret all the information from the genomic biomarker signature and to provide the hazard of a specific test item. So I just wanted to cover these different building blocks or modules in a bit more in details. So when we talk about the biological system that is used in the GARD assay, we believe that the selection of a biological system is a really important feature in order to be able to construct a mechanistically based in vitro assay. So in terms of skin sensitization, the molecular mechanism being responsible for initiation of skin sensitization are today quite well described. So I try to summarize some of the most important key features within this figure. So it basically all starts with a chemical that penetrates the skin. It binds to endogenous protein to form a hapten protein complex. So this hapten protein complex is then recognized by dendritic cells, which in response to different type of inflammatory mediators migrates to the draining lymph node, where it activates a T cell response and immunological memory. So each time that you're then exposed to the same substance, you will have a rapid proliferation of T cells that will give rise to a local inflammation in the skin, which is characterized clinically as allergic contact dermatitis. So in this process, we have selected to work with dendritic cells in the GARD skin assay, because dendritic cells is definitely one of the key players for developing this adverse outcome, which is characterized as skin sensitization. So, I mean, the hypothesis we had from the beginning was very simple. I mean, given the important role of dendritic cells for activation of skin sensitization, we believe that there should exist genes and pathways in these cells that were differentially regulated dependent on whether these cells were exposed to a skin sensitizer or a non-sensitizer. And of course, if we could identify this specific subset of genes, we can also use them as a predictive tool. So instead of just measuring one or two biomarkers, we measure basically the complete transcriptome of the cells in order to provide a more holistic readout of how these cells actually react to external stimuli. So that basically leads us to the training data set. So the substances in the training data set comprise a set of chemicals with known expected outcomes. They were essentially used in order to evaluate the hypothesis whether there would exist differentially regulated signals in the dendritic cells. So we used this subset of chemicals in order to expose the dendritic cells, and we basically measure the complete transcriptome of the cells using microarray technology. So for each of the chemicals, we measured a total of 29,000 genes per sample. So starting from the measurement of 29,000 genes per chemical, we were able to identify a subset of 200 genes that were differentially regulated dependent on whether the dendritic cells were exposed to a skin sensitizer or a non-sensitizer. And these 200 genes is what we collectively refer to as the GARD prediction signature, or the genomic biomarker signature. And this is also the information that is summarized in the heat map here to the right. So in order to better understand how these 200 genes relates to the biological relevance, we have also looked, of course, to the identity of the 200 genes and mapped them to the adverse outcome pathway for skin sensitization. So when we actually look at the identity of the 200 genes and we look at their involvement in different type of toxicity pathways, we can see that they are associated with genes that are typically involved in the upregulation of CD86 and other co-stimulatory molecules, but they're also involved in processes that are required for antigen recognition and innate immune activation. But they're also involved in processes that are typically referred to as key event 2 associated events, such as the protection against oxidative stress and the secretion of different type of pro-inflammatory cytokines. So in this process, this is typically what we refer to as being able to provide more mechanistic relevant information and to build an integrated testing system on itself. So in the final way, we have also defined a gene expression analysis tool that we use in order to interpret all this information from the biomarker signature. So in the final step, we basically use a machine learning algorithm that just compare the gene expression profile induced by an unknown test substance to the gene expression profile induced by skin sensitizers and non-sensitizers in the training dataset. So the important thing to remember here is that all of the 200 genes contributes to the final classification. The prediction model is fixed and it will not change. You will always have 100% reproducibility if you feed in the same data. And the interpretation of the results is really simple. So the prediction model uses a classification threshold corresponding to a decision value of 0. So a positive value will be classified by the algorithm as a sensitizer. A negative value will be classified as a non-sensitizer. So based on this overall process on how the GARD assay work, we have also managed to identify a standard operating procedure on how to test unknown substances. So this is just a brief summary on the conventional GARD skin assay. So the first thing we do is essentially to identify a concentration used for stimulation of the cells. And this concentration is essentially defined based on the cytotoxicity level of the test item. So in the conventional GARD skin assay, we perform the testing at one single concentration. So once the cell has been incubated with the test item, we isolate all the RNA material and we measure the 200 gene in the predictive biomarker signature. So in the final step, we basically use the machine learning algorithm in order to compare the gene expression profile induced by the unknown testing substance to the gene expression profile induced by sensitizers and non-sensitizers in the training dataset. And here you will also have the final classification as a skin sensitizer or a non-sensitizer. So that was a very brief and rapid introduction to the GARD skin assay and the concept behind the GARD technology assay. I just also want to say a few words about the GARD potency assay. So the GARD potency assay was developed using exactly the same 4 building blocks or modular components. And the GARD potency assay basically comprises of a complementary biomarker signature that can be used in order to further subcategorize skin sensitizers as 1A or 1B according to the potency classes in the CLP system. So we have also proposed that the intended use of the GARD potency will be within a tiered approach or a defined approach, where you basically use the GARD skin assay first in order to identify a skin sensitizer. And for those substances that are classified as skin sensitizer, you can then further proceed with testing in the GARD potency assay to further subcategorize the sensitizers as strong or weak sensitizers. So the first step in having in vitro assay accepted by regulatory units around the world is essentially to perform a formal validation study in compliance with OECD guidelines. And this is something that has been done for both the GARD skin and the GARD potency assay. So the figure here to the left basically summarizes the setup of this validation study that was performed as a ring trial exercise. So testing was performed in 3 laboratories. And in the first step of this validation phase, a total of 28 substances were tested in the GARD skin assay. And in the second phase, further testing was performed on the identified skin sensitizers in the validation phase 1. So based on the results here, we can see the column furthest to the left in both this figure represent the expected classification based on the reference data. And here to the right, you can see the classification from the different labs. So you can see that both the GARD skin and the GARD potency model is very reproducible between laboratories. And when we could evaluate the predictive performance of these assays, we can see that the GARD skin assay reached an accuracy of 94%. And when you use the GARD skin assay in combination with the GARD potency assay for potency predictions, you can have a predictive performance of 86%. So all this data has also been submitted to regulatory bodies, and we are now finally waiting for the final OECD approval. So this data has also been published in peer-reviewed scientific journals, and you can see the reference to the different publication in the bottom left of this figure. So that leads me also to the GARD skin dose response assay. So the GARD skin dose response assay is essentially just what it sounds like. It's basically the GARD skin assay performed in dose response measurements. So the way we propose this assay to being used is essentially to perform the GARD skin assay in a tighter range of different concentration. And we then basically just apply the conventional GARD skin protocol in order to generate decision value for each concentration. So the concentration can then be plotted versus the GARD decision value, which in this case is the response value. And you can generate different type of dose response figures. So you can see one of the dose response figure here to the right. And the readout that we're looking for in this specific assay is not only a yes or no hazard classification, but we're looking at the concentration, the lowest concentration required in order to give rise to a positive classification in the conventional GARD skin assay. So that corresponds to the concentration required in order to basically give rise to a positive decision value, which is the classification threshold for a positive classification. And this concentration is referred to as the CDV0 value. And for those of you that are familiar with the local lymph node assay, you can see that these response values that are measured in the GARD skin dose response assay is basically just an analog to how the local lymph node assay work. So we have evaluated this GARD skin dose response protocol using external data. So this was the initial data set we generated for a total of 29 substances. We can see the corresponding dose response figures to the right in a summary plot here. And in this specific case, we colored the chemicals based on their CLP categorization. So the pink color here corresponds to the strong sensitizers in class 1A, and the blue one corresponds to the weaker sensitizers in 1B. And what we can see here is that there is definitely some kind of potency information, and the strong sensitizers typically induce a positive CDV0 value at a much lower concentration than the weaker sensitizers. And from this figure, we can also see that there is almost a complete separation of the different CLP categories. But it's of course also interesting to see how this assay actually performed on a continuous scale. So the way to evaluate this is of course to compare the generated CDV0 values for these substances versus a continuous potency metrics. And in these cases, we use the local lymph node assay EC3 values. So when we correlate this CDV0 values obtained in the GARD skin assay versus local lymph node assay EC3 values, we could observe a strong and positive correlation with a Pearson correlation coefficient of 0.81. So there is a very good correlation between the CDV0 concentration obtained in the GARD skin assay and the local lymph node assay EC3 value. And another quite interesting thing to put on mind here is that also the pre and the pro-haptens fit the same regression line, indicating that this assay would be able to actually pick up signals also from these type of substances that would require some additional type of activation in order to act as viable sensitizers. So in terms of skin sensitization, we are also quite fortunate because we don't need to only compare the data to animal data because there's so much human data also available in the scientific literature. So we did the same correlation and looked for a correlation between the CDV0 values in the GARD skin assay versus the human NOEL values identified from the scientific literature. And also here we could see that there was a strong correlation between the CDV0 values in the GARD skin assay and the human NOEL values with a correlation coefficient of 74. So based on these observed correlations between the CDV0 values obtained in the GARD skin assay and the different potency metrics, we have also defined a protocol or a workflow basically on how to use this information to predict potency also of unknown test substances. So the way we propose this to be used is essentially to first use the GARD assay in order to derive a CDV0 concentration. And you would do this in order to perform testing in a titrated range of different concentration, and you would identify that CDV0 concentration based on the dose response curve. So in this specific example, we show a dose response curve from the chemical resourcinol, and here we could identify CDV0 value of 109 micromolar. So the way this information is then used in order to predict potency is essentially to use this experimentally derived CDV0 concentration in order to predict a corresponding EC3 value or human NOEL value using a linear regression model where we basically exploit the previously observed linear correlation between the CDV0 and the EC3 or NOEL value. So taking the same example here again, we can use the CDV0 value for resourcinol, and we can use that input into the linear regression models in order to predict a corresponding EC3 value. So in this case, for the resourcinol, the predicted EC3 value was 11.3%, which is very close to the observed value from the local lymph node assay. So having now also introduced the concept of the GARD skin dose response assay, I would also like to present a user case or a case study from one of our recent client project where we use this dose response assay in order to predict a corresponding local lymph node assay value that were then further used for risk assessment. So in this specific case study, we were approached by a client that were interested in potency data for a total of 4 test items. In this specific project, they were also unable to use animal models such as the conventional local lymph node assay, since the ingredients were generally intended for different types of cosmetic applications. So the way we approached this project was essentially that we initially take into consideration that the customer has already provided us information that this might be skin sensitizers, all of these test items. So we started this project with performing the conventional GARD skin assay. And for test items that were then classified as skin sensitizer, we proceeded with the dose response measurement where we tested in a titrated range of different concentration. So after that, we used the CDV0 concentrations as input value into the linear regression model, which were used then in order to predict a corresponding local lymph node assay EC3 value. We can also, based on the linear correlation plot, predict a 95% confidence interval for these predictions. So having a look at the results from this testing, you can see the results from the binary hazard classification in the figure to the left. And you can also see that all of these test items were predicted as skin sensitizers, as identified by their positive decision values. So we proceeded with testing of all these substances also in the GARD skin dose response assay. And these are the figures here to the right. And you can see that all of these test items gave rise to quite nice monotonic dose response curves. And we use this information in order to identify the CDV0 value for each of the substances. So these observed CDV0 values were then used in order to predict the corresponding EC3 values. And we can also predict the 95% confidence interval in order to also have a look at the uncertainty in the predictions. So the predicted EC3 values can then, of course, be used with a similar weight as a conventional local lymph node assay values. You can use them either for subcategorization into CLP categories, and you can also use them for further subcategorization into the SETOC categories. But you can also, of course, use them as input into already established methods for quantitative risk assessment, such as those defined for the fragrance molecules, for example. So this was just a final case study to demonstrate how this assay can also be used within R&D projects. So we are now approaching the end of this webinar. So I just wanted to provide a very brief summary of the main take-home messages from this presentation. So both the GARD skin and the GARD potency assays has been defined as OECD, included in the OECD test guideline program. They have an OECD test guideline program number. And they are also now accepted for regulatory studies, such as the REACH registrations within a weight of evidence approach. And the GARD-defined approach, where you use a combination of GARD skin and GARD potency, offers a unique opportunity for potency classifications according to the CLP classification system. And the GARD skin dose response is a brand new method for a quantitative potency assessment. And the assay may be useful for ranking of candidates based on skin sensitizing potency, but they can also be used to predict a corresponding local lymph node assay EC3 value or a human Noel value. And it has not been the topic of today's presentation, but we have quite a lot of data indicating that these testing strategies are also applicable for testing of more complex substances without a defined molecular weight, such as UVCB mixtures and different type of formulations. So by that, I hope you have got a better understanding how the GARD assay work and how the GARD skin dose response assay can also be used for quantitative risk assessment. So by that, I would just like to end this webinar and thank you all very much for taking the time to listen. And I will be really, really happy to take any questions or comments. Thank you very much.